There you are. You are there. Although you have a very large insect behind you. I don't know if you know that, Rick.
I thought I got them all last night. One back there?
He's still back there.
Wow.
But I hope that does not deter our audience. We are happy to welcome you here today. My name is Ann Merchant, and I work for the National Academy of Sciences. And Rick, as he is sitting with his insect.
I'm Rick Levert. I'm the program director of the Science and Entertainment Exchange, a program of the National Academy of Sciences.
Yes, and of course, I generally try to start by talking a little bit about the work of the Academy. And for the second event in a row, I've sort of come up empty with how much we have that's relevant to today's event. We do have some things. We have an older report. It was actually very important at the time, but I think it dates back to 2007 on the importance of pollinators in the ecosystem. And our division on Earth and Life Studies does do things that relate to the insect population and climate change and things of that sort. So I think Sachi is going to drop a link in the chat to that division at the academies, because that's where that relevant work would be done. But we don't actually do a lot on insects per se, but that doesn't mean that we can't touch on that kind of topic in the way that we put our events together for the Science and Entertainment Exchange. In fact, that's part of why the exchange exists, to think about how we consider the topics in science and go a little bit more broadly than the academies might do in the reports that they write. And so, of course, I think that's a good segue to you, Rick, to talk a little bit about the exchange.
Sounds like my cue. So if you are a writer, producer, director, studio executive, storyteller in any medium, a novelist, and you have a question about science as you are developing a story, you can call us and we will connect you to someone who knows what they're talking about and can answer your questions and put them in context of your story. We've done this over 3,500 times, including many movies, virtually every film that Marvel's put out since Iron Man 2 as an example. So if you are a filmmaker or a storyteller in any form and you have a question, please do call us. We are happy to answer those questions. If you are a STEM professional and you're just hearing about our mission, please do write us. These events have brought in so many new volunteers who are interested in being connected, and we're always happy to connect you to storytellers. You can find a recording of this show after the fact on the Exchange's website, or you can subscribe to the Science and Entertainment Exchange on YouTube to see these events and other videos like our Ask a Scientist series. So you can also sign up on our website for our Collider newsletter, which is a weekly newsletter of interesting stories in science. Well, we think they're interesting anyway. And when the video is available, everyone who is currently RSVP'd will get an email, so you'll be alerted. It should be later this week. I want to thank our sponsors, Howard Hughes Medical Institute, without whose help we would not be able to do these events. So thank you so much. We also get major funding from the Alfred P. Sloan Foundation. And I want to thank all the donors, especially individual donors who gave to this event today. Thank you. We couldn't do these events without you. We really do appreciate that. Today, you are going to hear from two incredible people from the Natural History Museum at the Smithsonian. They will be in conversation. If at any point you have a question, you can put it either down here or up here, depending on what you're watching us on. And I will be passing along as many as we can get to our conversationalists, to Carla. If we don't get to your question, I'm sorry. It was me. It was not Carla. And so that gets me to my rabbit hole of this week. My rabbit hole is an article I found on why farming and climate are contributing to a mass die-off in bugs. And that was depressing. So you are welcome to read it if you would like, but don't expect it to be happy. So Ann, what's your rabbit hole this week?
Well, mine is much happier, actually. It's a book that I've been reading. It's called Pilgrim at Tinker Creek by Annie Dillard. It is not a new book. This was published some time ago, but it is a nonfiction book, and Annie Dillard spent a couple of seasons around Roanoke, Virginia, where she really just studied her environment really carefully, and some things that Matt talked about when we were getting ready for this event, really just about spending time in your backyard, where she really looks carefully at what's out there when she would go for her hikes and her walks. And talks at great length about the butterflies and the grasshoppers and the dragonflies and all of the different things that she sees, including spiders, which are arachnids and not insects in the same way, but it's a really lovely meditation on what you can see if you look carefully. So it's not depressing. It's actually very interesting. So that's what I recommend. But I think that we can hear from our expert today, Matt Buffington, who I do want to say that we have to give special thanks to Carla Easter, of course, who's going to facilitate and moderate today's conversation, because when we were sitting and thinking about, because the Academy doesn't have a lot of work in this area, we thought, oh, who do we know? Who can we call? And of course, the first name that both of us that came up with was, we have to call Carla, because of course, the National Museum of Natural History has this wonderful insect zoo that I have spent many hours in over the years. And we knew that Carla would know just the right person to reach out to, which of course she did. And so it's always nice to know people who know people. And that's how we sort of networked our way to Matt, who we immediately agreed was exactly the right person to do this with. So we would like to ask both Carla and Matt to turn on their cameras so that Rick and I can go away and we can hand this conversation over to the two of you, which is why people are here, not to hear us, but to hear from the two of you. So, and of course, Matt, the thing that we thought was so great was we knew that murder hornets, which we just love to talk about, like that's your jam. So we hope that you're going to talk at some point about murder hornets. Okay. All right, great. I'm buzzing away and leaving it to you.
All right. So Matt, well, first let me thank Matt. I'm so excited about his presentation. We had a wonderful discussion ahead of in our preparatory talks. And I just am really excited to hear what he has to talk about. I mean, he's much more complex than just murder hornets. So I'm going to just throw it to him and we'll get started. And if it's okay with everybody, I'm just going to interject some thoughts, some questions. And as Rick said, if the audience has questions, you can drop them either up or down, depending on where you are, and we'll try to get to them. So go ahead, Matt, you take it away.
Cool. Well, thank you so much for inviting me. This is a real honor to be able to speak to such a diverse audience. I'm very keen on scientific education and scientific communication. I believe the work that members of our laboratory do here is absolutely fascinating. And the more people that know about it, the more support we can get and behind some of our initiatives. So I am going to share my screen. Well, but maybe before that, I'll kind of introduce the laboratory. So I work for the U.S. Department of Agriculture. Within that department, we have the Agricultural Research Service. We're the research branch of the USDA, and we do all the research for anything for preserving foods to growing better wheat, stronger chickens, and in our case, the systematic entomology lab. We're the ones that do the identifications and research and naming of insect species that may be part of our natural or agricultural ecosystems that may be of concern to us. There are 17 of us in this laboratory doing everything from scale insects to weevils to flies to mites, even outside of insects, and everything in between. We are stationed in the National Museum of Natural History, which is where we are right now. This is my office. So the collaboration between the USDA and the Smithsonian, I have heard from numerous higher ups, is one of the last great ideas in US government to join two major departments in one collaboration, and that is to make the National Insect Collection, which I'll show you some pictures of today. I consider the National Insect Collection a national treasure of the United States. It's one of my pride and joys to work on it, to take care of it, to curate it, to make sure it's taken care of very well, to loan specimens, to host people to look at it. It is not for public consumption. It is behind doors. You have to get an invitation or request access. It's public in that way. It's not out for everyone else to see. And it's a three-dimensional database through time and space of all insects that we have ever found in this country and countries outside the United States that we may do a lot of trade with, that we might have concerns with, that we might be worried about, or we're just plain fascinated and we want to know more about them. So with that little intro, I'll start my screen here, and I just have some slides that are kind of like talking points, nothing really, we're going to be kind of fluid here, but this, for those of you who have not visited Washington, D.C. in some time, this is the National Museum of Natural History, and I'm located behind some of the windows there on the right inside of a courtyard that has been expanded. Okay, so this is kind of what brings us to this beginning of this conversation today. This is a species we call Vespa Mandarinia, the Asian giant hornet, also called the murder hornet. There are about 9 different common names for this particular species. It's so big and in your face, literally. Things like this get a lot of attention. Now, we're concerned about this species as folks in Asia are concerned about this species, is that they are specialist predators of social insects, in particular, honeybees. They will decimate a colony, killing all the workers by decapitation. Very few Vespa Mandarinia are required to pull this off. And then once all the workers are done, they take all the larvae, which are living in the hive, and feed their young with those larvae. So in a way, they're kind of just doing their thing. They're trying to get through life like the rest of us. It just so happens they attack something that, 1, makes us honey, 2, pollinates our crops. Interestingly enough, they're not as big of a problem in Asia, where the bee that they use there, Apis certina, is actually co-evolved with this particular hornet. And they have a strategy. They essentially pile on top of a scout when it enters their hive, and they cook it to death with beating their wings and moving their muscles. And they literally kill the scout before the scout can report back to its nestmates where this protein source is. Now, Apis mellifera, which is the honeybee we have in the United States, does not have this strategy. They did not co-evolve with this hornet, and so they're very susceptible. Now, some consider this non-native Vespa here, Vespa Mandarinia, possibly a biological control agent of Apis mellifera, itself not native to the United States, and somewhat problematic when it comes to pollinating native plants. They sometimes get in the way of our native pollinating bees, which we have the most, where the highest species richness of bees in the world are in the United States. Little known fact, take that to the next cocktail party. That'll get people talking.
So Matt, can I ask a question about that?
Absolutely. Sure.
So I'm fascinated as you saw in the beginning. I've become all into honeybees right now. It's kind of my thing. So again, maybe you can talk a little bit more. It's fascinating to me that our honeybees have not co-evolved or have not been able to find a strategy for this. And I guess maybe I'm trying to visualize, is the major effect of the murder hornet on what we think of as industrialized kind of honey? Or if you were in the natural, like if you happen to overturn a trunk and you see a natural hive, right? Are those honeybees more likely to be able to ward off this kind of attack rather than ones that we think about more industrialized? Basically, the whole goal is for them to create honey for us.
Yeah, that's a neat question. I like that. My answer, let me couch it in the fact that I'm not sure we really know the real answer to that question, but it has less to do with the industrialized organization of hives versus a wild hive, if you can imagine that. It has more to do with the behavior of Apis mellifera. That's as long as it's the same species, wild versus in a nest box. My guess is they're both susceptible the same way. They simply don't recognize when one of these scouts of Vespa Mandarinia enters their nest. They don't recognize them as the threat that they are. So they really don't know what's going on until the hornets come back for more and they start taking larvae. Then they raise the alarm. But when one's just cruising around, it's kind of like they miss it.
That's fascinating. Because, you think about the various types of honeybees, some that are more aggressive than others.
Yeah.
But you think they would have been better at recognizing this. That's fascinating.
And you bring up a good question. I mean, there was this Africanized honeybee.
Mm-hmm.
I don't know how they respond. I don't know if anyone knows how Africanized honeybee would respond to Vespa Mandarinia attack. Right.
Fascinating. So we do have a question. Ida asks, how did this hornet come to this continent?
Ooh, the $64,000 question. If you guys remember that TV show, that's exactly what we're trying to find out. So we have commerce with Asia all day and all night, coming into 15, 20 ports coming from British Columbia all the way to Baja California. Trying to inspect all that cargo and look for things on that cargo that we don't want in our country is the job of Customs and Border Patrol, as well as APHIS, the Animal Plant Health Inspection Service, which is the kind of law enforcement branch of the USDA. So there's all these containers, there's lots of cargo coming in. Our guess is that this thing just slipped in. Like it was basically, it didn't come here like on purpose, like, I'm going to invade the United States. It's more like, hey, this looks like a cool place to hang out. I'll hang out here for a little while. Next thing you know, it's like in the middle of the ocean. It's cold. Who knows? It just kind of hunkers down, and then it makes landfall, and it starts to go about its way. Now we do know the climate around the Seattle area where these were discovered is consistent with the climate of their native range, say in northern Japan, parts of China. So they're happy there. Where I did my degree at UC Riverside, I don't know if they can handle it there. It's pretty dry. It's pretty hot. The whole LA basin may not be a great place, though it's artificially wet because of the irrigation. So, these are very interesting questions, and we're actually probably too early in the game to get an idea of how far they could go. But we're also working with our partners all over Asia to look at the genomes of various populations to try to triangulate where in Asia they came from, the populations that we have in the United States.
Okay. So the questions just keep coming. I'm going to combine two. So actually, they're separate questions, but I'll put them into one. So first, if it's possible for praise to develop a defense, how long would that take in terms of evolutionary time? And then a question about what is the hornet doing in the picture you have up? Okay.
Evolutionary time. Even insects with very rapid reproductive rates, we would be looking at quite some time here to evolve the behavior that's in the Asian species of bee that is able to kind of dogpile, for lack of a better term, and kill the scout. That's a very complex behavior to learn how to do. You can evolve resistance to pesticides in a few populations, especially the way broad spectrum pesticides are utilized in the United States. So that kind of evolution happens very fast. And basically you're killing 999,000 individuals and maybe 100 survive because they've evolved a particular trait to detoxify. Those 100 become the basis of your next generation. And in very few generations, you've got resistance. That's a very simple type of evolutionary trait versus a behavioral trait, in this case, a little bit more complicated. But it's a good question. I don't have a good answer for it, but it's certainly something that's something I've thought about. How long would it take Apis mellifera to develop that instinct? Okay, so in terms of what these wasps are doing, well, one's just unhappy being held to show kind of how big they are. That's a queen. The queens are bigger than workers. The workers are pretty big too. The one that's eating there, this is a fascinating picture. This is one of those pictures that is gonna, I hope, be very famous in the future. So what this picture is the hornet that was radio tagged. That's what that transmitter thing is coming off that antenna. That's a radio tag that they used to track back to where the nest was, what we're calling nest zero. They used a radio transmitter and followed this hornet back to its nest. We weren't sure where it nested. And all the sentinel traps that were run killed the hornets. So you would say like, okay, we know the hornet's in this county, but we don't know what it's doing. Well, the USDA developed the lure and worked with U.S. Department of U.S. Department of Agriculture in Washington to tag this thing while it was still alive and then track it down. So it was a nice collaboration between state and federal agencies, and they nailed it. They found the nest. Like, that's cool. And we got an idea of how big the nest was. They were ready to go, like spread to create new nests. Like, the timing was unbelievable. And just as a capstone for this story, the nest is actually going to go on display here at Natural History in July. That's just as a kind of a cool thing, you know?
That's very cool. Very cool. So Quaylen asked, oh, this is fascinating. Would hybridizing native bee species with Chinese bees allow the native bees to better respond? I guess this question about the behavioral, could you somehow genetically, bring in those behavioral traits, that would take much longer to develop if we didn't find some other way to introduce them?
Yeah, that's like that's a very likely solution right there. That's a very good idea. If you talk to beekeepers in Asia, they simply just like put screens up over their hives that are big enough for the bees to get out, but too small for the hornets to get in. That's kind of the easiest solution. But so I think that's probably the route people will go, or let's have some more awareness. So it's one of these things like, do you want to start a breeding program with hybridizing bees? Possibly the Africanized honeybee is a good example of, trying to make a more efficient pollinator, ended up making kind of an angry pollinator. And that kind of went the wrong way. Like that wasn't expected. You're kind of monkeying things around a little bit when you're crossing things that can sting. So it's possible, but I don't know if you want to do it. There might be easier solutions.
Yeah. So
like put screen over your nest.
Yeah. As speaking as a budding beekeeper, let's do the screens, because again, if you mess up the behavior, right? Yeah. Africanized honeybees are great honey producers, but nobody wants to work with them, right? Nobody likes to get stung that much. So I'm with you. We'll just put the screens over the hives and hope for the best.
You can also I've heard you can also work on your backhand too. That using a tennis racket is a perfectly adequate way to protect your hives.
Yeah.
Okay.
Andy.
Anything else on this particular?
Nope. I think we're good for questions. If you want to go ahead, we can move on.
I'm trying to remember what I've got here. So this is kind of my process. This is how we do things here. So the drawer there on the bottom left, that's how we store our insects. That's how we store specimens here at Natural History, and how most insect collections, what they look like. They have different dimensions. They use different sizes of things, but this is how we organize. And this is what I use when the specimen came in to me, the first one that was caught on some guy's porch outside Seattle. It was FedEx to me, and with a lot of concern, you just double check that if this is right. We think it's Vespa Mandarinia. Can you double check? And that was kind of my job. Let's confirm this. And so I went into the collection. I went in to see and sure enough, I'm like, yeah, this looks like Vespa Mandarinia. What blew me away, that unit, that box at the bottom right of that drawer there, which has the big label in it, that was the only specimen from North America in this whole collection. All the rest were from various parts of Asia. And that's the first light that went off, I have a feeling this is going to have some pretty big ramifications when I put my final ID into the database. And sure enough, it was pretty almost instantaneous that this was a big threat. So this just gives you an idea. This is the specimen, the one there with the ruler behind it, those are in centimeters there, so you get an idea how big it is. It's big. And this was a queen. And this was the first specimen I saw that kind of started things off here. So this is how we, imagine this drawer times 275,000. I think that's how many we have here in natural history. So it's a very big collection. And this is how you reference what you're looking at. If you don't have a collection, you can't confirm what you have in front of you is what you think it is. And this brings up some very interesting questions. Who's to say Vespa Mandarinia doesn't belong in the United States? Well, it's not like a rule, but we have thorough collections of all the hornets of basically the northern hemisphere, and we know where they naturally belong. So we have a Vespa crabro here in the United States. That's the European hornet. Doesn't really do much. It's a cool thing to have around your garden. It kills things that eat your lettuce and stuff like that. But it's not a threat. This particular thing was a threat because it obviously has an impact on apiculture. But we wouldn't know that it didn't belong here if we didn't have previous collections already in that area and never found it. So it is this kind of chicken and egg scenario a little bit, but we did have lots of collections from Asia where it seemed to be fairly common, and it's a well-known apiculture problem there. And it kills a few people every year too from anaphylaxis. So was it not a new species in that respect, but we had a good 140 years of insect collecting in the United States, and we never came upon one until now. And it's the collection that allows us to make that decision. If we just threw everything away, we wouldn't have the reference. We wouldn't be able to recognize these things. Nature's too complicated. You can't just remember it. The collection is not only a document of the occurrence, but also it allows you to compare specimens and go, okay, this one's like a little orange. If you look at the head on this hornet, it's bright orange. Kind of like a football helmet, like Cleveland Browns maybe. That varies. That color varies actually quite a lot. And the more specimens you have from the more locations, it allows you to understand and triangulate about where that species is in terms of morphospace and color. These aren't really straightforward answers sometimes. So anyway, I think that's all I've got on this particular species.
So,
any questions?
Oh my gosh, you have lots of questions. They're piling up for you.
Okay.
So the first is, this is related to the last question we ended with, is does it make sense to use gene drives of murder hornets and/or release infertile females? And I was thinking about this. Basically, you'd have to be targeting just the queens, right?
Yeah, that's right.
Because they're the only ones that are able to produce any eggs. So. Yep.
Again, it does come down to what is the most effective means. Right now, the most effective means is actually monitoring and removal of nests. And if we can stay ahead of them, which I think we can, we're early enough in the game. If we get overwhelmed, like the floodgates open, then we're going to be looking at some of these much more technologically advanced methodologies for sure.
Got you. And then next question, has it been observed that the U.S. biological controls of other pest insects, such as spiders, birds, successfully subdue and eat these invasive exotic murder hornets?
Okay. No, not Vespa Mandarinia. We have no evidence of that. But I will talk about some successes and current projects in biological control programs in a minute, once we're done with this.
Right. And then let me just, I know we have a ton of questions, but I want to get you to some other things. So let me add, I think this question is really kind of cool. Can you tell from genomes, from genomic data, how many times these hornets have entered the US?
Oh, yeah, cool. Yes.
That's a cool question, right?
That's a cool question. So what we do is there's certain parts of the genome that are evolving very rapidly. Those parts of the genome that evolve rapidly allow us to distinguish at the population level like within reproductive units, who's related to who. So these are the gene groups that we focus on for these kinds of questions. And so using that, we can actually determine one, how many times has a species been introduced? Two, if we have their source populations, we know where they're from. And three, if we have that, we can figure out maybe how they're getting here. So that's exactly what we're trying to do. 100%.
So Matt, let me ask
what we're
you,
doing. Yeah.
Would you like to move on to the next slide, or I could ask you questions for the next 20 minutes if you like.
Let's move on, because I want to talk about some of the other kind of aspects of our lab.
Cool.
And we're going to go from these monster hornets, which, by the way, when they came, well, you'll see the kind of wasps I work on. When these came, I almost didn't know how to handle them. They were so big. Like they made me a little nervous, because what I normally work on are these. This is a United States dime, okay? These are 3 different wasp species they're totally unrelated to one another. They've been separated by, at least 80 million years of evolutionary time. The top one is this thing called Chalcodectus. I have no idea what it does. The bottom one is actually Preseucoila, this thing I named after Elvis Presley. The middle one is Trichogramma KK, and this little Trichogramma wasp, which is small enough to go into his ear and turn all the way around and come back out without ever touching the sides. This is one of the most potent biological control agents of various moth pests of wheat and apple and all kinds of stuff. So this is where I spend all of my time is on these very, very tiny organisms that happen to be so unbelievably speciose that we've named, at the best estimate, 5% of the actual diversity of planet Earth. And being so small, it kind of makes sense. Now, the other aspect of this is there's not many people to work on them. Look at all you normal people out there. Like, that's everyone. There's a tiny fraction that work on insects at all. And of those, it's an even smaller fraction that work on taxonomy, which is what we do here. If you look at how things are divided up across the board of what groups get all the attention, well, look at all the systematists is what I am. So look at all the systematists that work on vertebrates. That's anything with a backbone. The one insect group I work on is, which is these things called the fidgeted wasp. There are more fidgeted wasps, we estimate, there are more fidgeted wasps than all birds, fish, and mammals combined, and lizards and frogs and snakes and all those things. So it's a tiny family of very small wasps. So there's a lot of taxa. Those are species. That's in the red there on the right-hand side. And there's not that many people to work on them. So we form a very close association in systematic entomology, where you rarely find someone that you don't immediately have an instant connection with, because we're all there. Like, we're all dealing with this. And so it makes us a very strong community, a very well organized community. And we end up, it's also, if you're looking for a career where you're going to find the answer, this isn't it. Like, you're never going to find the answer. I'm never going to sort all this out. I'm going to die. And I know that there's there's stuff in my office that's still going to be here. Like, so you have to kind of triage things. So that's kind of the challenge. That's what I'm after. That's what I'm doing. This is one project I'm working on right now. The fly that's in the middle there, it's kind of orange, is sitting on a blueberry. Just to give you some scale, that's called the spotted winged Drosophila. A lot of folks are interested in fruit flies. They're used in labs. You can rear them. They're easy to take care of. They're genetic models. Well, this one, Drosophila suzukii, is the dreaded spotted winged Drosophila. And this thing is like a $500 million fly. Like, it causes so much damage worldwide. And it lays its eggs inside soft fruit. The larvae feed inside the soft fruit. You can't spray them. Larvae are inside. And, Mary's biting into her blueberry pie and there's larvae coming out of it. That's bad. Well, unless you're like entomophagy, but most people aren't. So the USDA is very interested in this, huge concern. It's native to Central Eastern China. It's also in Japan, Korea. So we do a lot of field work there. And it turns out this little wasp that's facing off against it there on the other side of that hole. This is Ganaspis brasiliensis. And this little wasp is going to be the focus of my work for the next few years in all kinds of ways, because it looks like it may not be one species, even though they all look exactly the same. There's some traits that we're trying to separate. But it's allowing us some insight into the biology of these wasps, which leads me to my next slide here. This is why I became a systematic entomologist that worked on these tiny wasps. So we call these wasps parasitoid wasps because they lay their eggs inside of the body cavity of another insect. And in the process of their development, eating the inside of the host, they eventually kill it. Parasites, on the other hand, have to keep their host alive. These must kill their host in order to complete their reproduction. So we call them parasitoids. We use a distinction there. How this is done is the stuff of legend and lore, and what brings me to work every single day. So if we focus in the center square here, you've got a wasp. She's got her ovipositor out. The stinger, by the way, in a hornet or a bee is a modified ovipositor. The ovipositor, or egg-laying device, kind of predates it evolutionarily. This device has been used by a lot of insects have ovipositors for placing their eggs in special places. Turns out parasitoid wasps lay their eggs inside of other larvae. First, they have to find them. How do you find something so small in this massive world we live in? That's one whole area of entomology, is how insects navigate. When you're this small, you actually swim through the air column. If you think about that, think of that little tiny trichogram I showed at the beginning. It's life at low Reynolds numbers. These things are struggling to get through the air column. They don't fly like a bird. They kind of struggle through it. There's great videos. Geez, if you want to get down a rabbit hole, look up trichogram of flight, and they've got some great high speed or slow motion stuff on YouTube. You can watch them how they move. It's amazing stuff. So okay, so first you've got to find your host. Good luck there. Okay, let's say you found it. Now you've got to decide, is it the right species? So the female lays her ovipositor into the host, and she's able to determine, what species is it? What stage is it at? Has someone else already been here and oviposited? And she can make a decision based on how long she's been alive and how many eggs she has in her metasoma. She can decide whether or not just to go for it. Like let's take a risk and hope they survive. She can decide to move on. Like she's early in life. She's got a few days to live still. She's going to bank on another host. So let's say she lays that egg. Well, now we've got two different fate pathways that have come in front of us. One is the host larva does not want to be parasitized. It's evolved resistance against parasitoid wasps. It has an immune system. It has little platelets that cover the egg. It's identified as foreign. And then as the larva develops and it molts to its next life stage, the dead egg is ejected out the cuticle and we're done. Well, the parasitoid wasps have also been co-evolving. And in this case, this little case here of Leptopolina, she actually has a viral genome inside her own genome. This is a mutualism that goes back millions of years that we don't quite understand. And this virus actually releases various proteins that block and neutralize the host immune system, allowing the parasitoid larva to continue to develop. In fact, in some cases, in some other groups, it convinces the host that the parasitoid larva is in fact itself, and it will take care of it, nurture it, make sure it's okay, like it's an organ. and it'll eventually kill it from the inside. This evolutionary ballet of death has been going on for millions and millions of years. Those are numbers I can't process how big a million is of anything. So trying to imagine how long something like this takes to develop, you kind of have to go outside of yourself to imagine that length of time. But this is why we use these in biological control, because of this specialization. She has, in this case, this Leptoplena actually cannot parasitize anything else. She cannot continue her lifespan on anything else except Drosophila zuzukii. So it gives us some security. She's not going to just go and attack something that we don't want. So that's where the taxonomy of these wasps comes down to me. There's, 80 species of Leptoplena. They all pretty much look the same. They're all pretty tiny. It's up to me and some of my former students, some of my colleagues. We work out how to tell these things apart, and we try to provide characters so non-specialists can have a wasp in front of them and figure out what it is. What are they looking at? We are getting into the stage, though, like with Vespa Mandarinia, where we're looking at genomes. We're trying to look at the evolution. How did this viral insect mutualism evolve? That's what a genome is for. We'll be able to at least have a better idea once we exploit that. So the life cycle on the right hand side, that you can see basically the pre-pupal adult is hiding out inside the former fly case. That's what you get, and that's what you want when you're trying to control something like this pest. And as a result, I am conservation minded, and I do like to see the reduction of the reliance on pesticides. And this is one way to do it. But it's very clear the early stages of biocontrol, we're not interested in the green movement. It was actually the only way to control the pest. We came from an era of using heavy metals and arsenic, kerosene, you name it, to try to kill things. And it turned out that finding the natural enemy, as we call it, to its proper host and making that match is much more efficient at killing your pest and your non-native. So anyway, this is where I spend all my time, is figuring out not so much these life cycles, but how to tell the different wasps apart from one another. We have any new questions at this moment?
Oh my goodness, I have lots of questions, but yes, you have some questions.
Right.
So let me figure out, where should we start with? So there was a previous question before that I'm going to link to a question that Mary asked, and there was a question about citizen science. Excuse me. And again, are there spaces? Donna asked, how important is the role of citizen scientists to being able to, her question was about putting out traps and IDing things, but I'm thinking much larger. How can citizen scientists help in the work that you're doing? And then that leads to another question about, do you take volunteers in your lab?
So basically, citizen science is how we're going to make progress on these questions. There's just not enough of us. Like I showed, that pie chart's kind of a joke, but it's actually not a joke. That's serious. We rely on citizen science every day for the work we do. In fact, the Vespa Mandarinia question, that's a citizen science success story, because the lures that are put out, that's all citizen science. My buddy Chris Looney, who runs that program out there, is just unbelievable at his ability to get people motivated. I have another friend, Sam Droege at U.S. Geological Survey, Pollination Lab. He gets people to survey for native bees constantly. They're just like, he has this energy and this enthusiasm that gets people just so into it. With parasitoids, it's a little bit trickier in that, not that it's not possible, the taxonomy of parasitoids is so much more difficult and requires more specialized equipment. I'm looking at my microscope right now, like that's like a $19,000 piece of equipment. And that's kind of like entry level to get the right kind of optics for making these kinds of distinctions. However, that doesn't mean it's not possible. We take interns and volunteers here at Natural History. We're working through that program right now. There's some changes that are going to be taking place. But the goal is to not turn people into systematic entomologists, but I want people to at least leave our lab with some understanding of how the natural ecosystems work. And if you can get people at the college level, they take that with them. They become the voting public. They become the ones making the decisions. My daughter's scout troop, same way. This is the next group of people. And again, they don't need to become systematic entomologists, but it's good that they understand there's orders of insects, and some of them are, most of them won't hurt you, and they're doing ecosystem services. So citizen science is very, very important. If you don't have iNaturalist, that's like the gateway drug to becoming citizen science. Like it's an incredible resource. We use it a lot, for sure. My wife studies weevils. She's found new state records, new county records. Like we've actually figured out where to go collect them because someone posted a picture from their phone.
Yeah.
You know?
iNaturalist, I'm with you totally. That is like the gateway to it.
Yep.
So Robert asks, is sterilization / release of male insect species an effective strategy for pest control?
Yeah, I think for some groups it is. I think that's what we've been doing for what we call the Mediterranean fruit fly, Ceratitis capitata. So there's certain groups where that is a very effective means. I think they're exploring that with mosquitoes. There's a number of groups where you can irradiate a very, very large number very quickly and let them go. You know, it's one of those, again, it's a logistics thing of now that they're here, it's clearly not getting that genie back in the bottle. So that's a very effective way to deal with certain, it depends on their reproductive strategies. I won't even get going in that with Hymenoptera. They're so weird. So, but a fly is very straightforward, and that's a very effective way to do it.
Right. And then Susan asked, how does the ability to disengage the host immune system cross over to human viruses?
Oh yeah. That's this is something that we've discussed a lot within entomology discussion groups around the coffee urn. We just don't have a lot of medical people like hanging out with us. But it seems like the one thing to remember, or not remember, to consider, is that one of the reasons why insects are so bizarre to us, while they're terrestrial like we are, we diverged from them 300 million years ago. Like we do not share a recent evolutionary history whatsoever with them. So some of the things that operate in them may not operate in us. However, there's a person in my lab that helped understand there's a naturally occurring screw in beetles that's part of their leg system, totally independently derived from humans inventing the screw. Most drones are based on flight technology derived from insects and how insects fly, because insects fly very differently than birds. Their exoskeleton is almost mechanical in a way. There are lessons to be learned and information to be gathered that apply outside of entomology to real world problems. And I would love to see more of that kind of work. And it's definitely the direction some labs are going. I don't know what the current status is, but if you can mask something, if a viral genome can produce something that masks something to basically hide out inside something else, man, you've got to figure that one out. Like at the mechanical level, like how could we do that? Because it also means you could do the opposite. You could highlight something, like we do with vaccines, but maybe we could do it more efficiently.
Absolutely. I mean, I guess for me, fundamentally, I think there's this, most people don't realize that insects have immune systems.
Right.
However rudimentary they may be, they have developed an immunity to particular things. So again, there is, although it may not be as complex as a human system, it obviously, there's, something that works in evolution just continues to thrive. So I also find that absolutely fascinating.
Yeah, and it begs the question in the case of the movie franchise Aliens, that's a parasitoid The alien is a parasitoid. Humans are the host. How is the alien overcoming the host immune system? You know, did they poison them? Because that's another way you can some parasitoid wasps lay their eggs in the brain of their host where the immune system's not as strong. So they'll hide them. They'll hide their eggs in certain body parts. And that's kind of fun and it's kind of bizarre, but this gets really amazing in that the parasitoid does need certain nutrients in order to develop. If the host isn't consuming them, the parasitoid can direct the host to go somewhere it normally wouldn't go in order to get those nutrients. So it becomes essentially zombified, whatever that means. Basically, becomes an automaton, and the parasitoid's at the helm, at the controls. That's probably the strangest of them all. As you know, George Gaylord Simpson, evolutionist in the 30s, was fond of saying, you know, evolution's nature is actually beyond our ability to comprehend how complicated it is. Like we can think we understand it. Our brains actually can't. Like, we're not there, you know. All right, should I move on?
Yeah, go right ahead.
So this is a biological control system that we're working on right now. We use these gigantic, what we call phylograms or evolutionary trees. This is derived from genomic data. We run them through supercomputers to find out how things are related to each other. This is a kind of roadmap of how we think things are related to one another. We use this in a predictive context to understand who might attack whom and when. So this is another aspect of my work that I do, is building these big trees. This is another very tiny wasp. This is actually much smaller than the one we were just looking at. Her name is Trisulcus japonicus, otherwise known as the samurai wasp. This is a very successful biological control story happening actually right now. She is an egg parasitoid. So Ganaspis brasiliensis that we were just looking at before eats maggots, has to attack the larva. Well, these are stink bug egg parasitoids. If you live on the East Coast, stink bugs are a problem. It's an Asian, non-native, very invasive species that is a destroyer of crops, competent on 200 different kinds of plants. I've actually met farmers at extension events, almost in tears, that their farms that have been active for 7 generations are done because of this thing called the brown marmorated stink bug. That's very serious. And I take my job very seriously, trying to understand how we could help. What can we do? You can't just spray broad spectrum pesticide all over the East Coast, killing absolutely everything because you've got this stink bug. I guess you could. It would be very irresponsible. And it wouldn't work, by the way, because the stink bug would evolve resistance. So we look for parasitoids. These also come from Asia. And these are actually quite, first of all, they're adorable. I mean, you can make a plush toy out of this. Second, they attack the egg stage, meaning the nymph of the stink bug never even has a chance to start feeding. So it's the most efficient kind of parasitoid you can find to attack a pest are egg parasites. This is an astounding photo, by the way, taken by my former postdoc, Elijah Talamus. So this is the life cycle of the so-called samurai wasp. So these eggs also have an immune system. That was like news to me. Like, I was like well into my career here when I found out eggs have immune systems. So don't tell my supervisor. But if the eggs basically go unattacked, you get your stink bugs eating everything from soybean to pumpkin to oak trees to apple. But if the parasitoids get their way, they will take the place of the nymph. The female, it's really cute. She'll lay her egg in a clutch of eggs. She'll attack every one of them, and then she'll sit on top and defend it against a competing female. Because if you get more than one parasitoid in a host, one might kill the other one inside the host. They both may die. All 3 may die, including the host. It's kind of it's a bit of a mess. So she defends the egg mass to ensure that her own genes are the ones that move forward. So this is part of an outreach poster we did in order to help educate other labs on what we think is going on here.
So Matt, I am. Oh, that was your last slide.
That's the end of my slides. So we can go back and forth. We can do all kinds of stuff there.
Almost perfect. Well, we have about a minute.
Oh boy.
Left before you were perfect timing. So in about a minute, maybe you can give your last thoughts before we turn it over to Anne and Rick. But I want to tell you, thank you. This was fascinating. I'm sorry we didn't have enough time to talk more about the stink bugs and the samurai wasps, because that's awesome. But I'll let you go ahead and have the last word.
Yeah, I think I want everyone to kind of take a moment to consider their environment. Insects, these natural enemies, these parasitoids, all the insects that we encounter are such important parts of our ecosystems and agricultural systems. I am very concerned with what we're seeing as such a huge die-off of insects. And while a lot of people think it's funny or amusing or even a solution to being annoyed by insects, it's something I think we really need to be very careful with. Insects are being they're a canary in the coal mine right now. And if they can't survive, if we're killing them, if we're destroying their habitat, it's not going to be long until we're in big trouble too. So and also they're fascinating. They're fascinating animals. They don't have an advocate. They don't have a Bill of Rights. They are just trying to do their thing like the rest of us. And having some respect for that, I think you'll come to appreciate your garden, your park, your national parks, all of your habitats a little bit more. When you think about all these creatures out there just trying to make it, and how many are unknown. It's an unknown world out there. We don't necessarily need to go to Mars. There's like stuff to discover right here. And you just takes a little net. That's it. That's my soapbox.
Awesome. Well, I'm going to just end with a sentence that you told us. You said when we were preparing for this that insects are as annoyed with us as we are with them. So we cannot forget that. So with that, I'm going to turn it back over to Rick and Anne.
Well, I'm one of those people that does not kill insects, and I don't kill them in my house. I don't kill them outside. I do kill mosquitoes because they bite me, but otherwise I'm very, very careful about just trying to remove them and put them outside my house. So I take to heart what you're saying, Matt, and I do think that they're really interesting. And if you take a moment and just observe them, they can kind of entrance you because they're super cool and they're not like us. And who doesn't like to watch something that's not like us? So thank you so much, both of you, because Carla, you always ask such great questions because you're a naturally curious person, and we couldn't ask for more for somebody, as Rick said, in conversation with. And Matt, you did such a great job of answering, but you did introduce us to some pretty kooky things that, as I say, they're not like us, and maybe that's a good thing. But we're very happy that all of you joined us as well. And so Rick and I always want to thank everybody for being here. And when we have our next invitation ready, Sachi will make sure that shows up in your inbox, and we hope you will join us again when that time comes. So thank you.
Thanks, everybody.